Cells are the fundamental units of life, serving as the building blocks for all living organisms. Cells are classified into two main types, prokaryotes and eukaryotes. These two categories differ significantly in their structure and complexity. Prokaryotic cells are simpler and smaller, typically ranging from zero point 0.5 to 5 micrometers in size. They lack a nuclear membrane, so their genetic material, DNA, floats freely in the cytoplasm in a region called the nucleoid. Prokaryotes have few or no membrane-bound organelles. The two domains of prokaryotes are bacteria and archaea, which play crucial roles in ecosystems and human health. Eukaryotic cells are larger and more complex, typically ranging from 10 to 100 micrometers in size. Their defining feature is a true nucleus, enclosed by a nuclear membrane, also called the karyotheca. Eukaryotes contain various membrane-bound organelles that perform specialized functions. Examples of eukaryotic organisms include plants, animals, fungi, and protists. The cytoplasm is the material that fills both prokaryotic and eukaryotic cells. It consists of cytosol, a gel-like fluid, and contains various cytoplasmic organelles and structures. The cytoplasm serves as the medium for most cellular biochemical reactions and provides support for the cell's organelles. It's constantly moving, transporting materials throughout the cell. Let's compare the key differences between prokaryotic and eukaryotic cells. Prokaryotes are smaller, lack a true nucleus, and have circular DNA in the nucleoid region. Eukaryotes, on the other hand, are larger, contain a true nucleus with a nuclear membrane, and have linear DNA organized into chromosomes. Eukaryotes also contain numerous specialized membrane-bound organelles. Ribosomes are the protein synthesis machinery found in all living cells. Each ribosome consists of two subunits, a large subunit and a small subunit. Both are composed of ribosomal RNA and proteins. Ribosomes are found in both prokaryotic and eukaryotic cells. In prokaryotes, ribosomes float freely throughout the cell's cytoplasm. In eukaryotic cells, ribosomes can be found in several locations, free in the cytoplasm, attached to the rough endoplasmic reticulum, and even within organelles like mitochondria and chloroplasts. The endoplasmic reticulum, or ER, is a network of membranous channels found in eukaryotic cells, typically located near the nucleus. These interconnected membrane tubules and flattened sacs form a complex network throughout the cytoplasm, extending from the nuclear envelope. There are two types of endoplasmic reticulum: rough endoplasmic reticulum and smooth endoplasmic reticulum. The rough endoplasmic reticulum is characterized by the presence of ribosomes attached to its surface, giving it a rough appearance under an electron microscope. Its primary functions include producing proteins, storing and transporting these proteins, and adding chemical modifications to proteins. In contrast, the smooth endoplasmic reticulum lacks attached ribosomes, giving it a smooth appearance. The smooth ER is responsible for producing lipids and steroid hormones, detoxifying harmful substances, and storing calcium ions in muscle cells. Together, ribosomes and the endoplasmic reticulum form an essential protein production and processing system in eukaryotic cells. The Golgi complex, also called Golgi apparatus, consists of flattened, stacked sacs. The Golgi complex receives substances from the endoplasmic reticulum on its cis face, modifies them, and packages them for transport on its trans face. The Golgi complex functions like the cell's postal service, receiving, sorting, and directing substances to their required destinations. Additionally, the Golgi complex produces carbohydrates such as cellulose for plant cell walls and glycoproteins for secretion. Lysosomes are membrane-bound vesicles that contain powerful digestive enzymes. Lysosomes are formed by the Golgi complex, which packages digestive enzymes into vesicles that mature into lysosomes. Lysosomes serve as the digestive system of the cell, breaking down waste materials, foreign particles, and damaged cellular components. Lysosomes perform three main types of intracellular digestion. Heterophagy is the process of digesting substances that come from outside the cell, such as bacteria and foreign particles. Autophagy occurs when lysosomes digest the cell's own structures, like damaged or aged organelles. Finally, autolysis happens when lysosomes release their enzymes to digest the entire cell, which occurs during programmed cell death. Peroxisomes are specialized organelles found in eukaryotic cells. Peroxisomes participate in several crucial metabolic processes, including fatty acid and alcohol metabolism. Their most crucial function is degrading hydrogen peroxide, which is highly toxic to cells. Peroxisomes contain the enzyme catalase, which converts hydrogen peroxide into water and oxygen through the reaction, 2H2O2, 2H2O plus O2. Mitochondria are often called the powerhouse of the cell. They are unique organelles with a distinctive double membrane structure. Mitochondria have a distinctive structure with an outer membrane and a highly folded inner membrane called cristae, which creates a large surface area for energy production. Mitochondria are responsible for cellular respiration, generating most of the cell's energy in the form of ATP. Through this process, glucose and oxygen are converted into ATP, carbon dioxide, and water. What makes mitochondria truly unique is that they contain their own DNA, known as mitochondrial DNA or mtDNA. They also have their own ribosomes and can replicate independently of cell division. This independent DNA allows mitochondria to replicate on their own schedule, which is thought to be evidence of their evolutionary origin as once independent organisms. Plastids are specialized organelles found exclusively in plant and algal cells. Like mitochondria, plastids contain their own DNA and have the ability to self-replicate. There are multiple types of plastids, each with different functions in the cell. Chloroplasts are the most well-known plastids, responsible for photosynthesis and giving plants their green color. Chromoplasts store colorful pigments like carotenoids, giving fruits and flowers their yellow, orange, and red colors. Leucoplasts are colorless plastids that store nutrients like starch, oils, and proteins. Chloroplasts have a complex structure optimized for their role in photosynthesis. Like mitochondria, chloroplasts have a double membrane structure with an outer and inner membrane. Inside the chloroplast are membrane structures called thylakoids, often arranged in stacks. These contain chlorophyll, the green pigment that captures light energy. The fluid-filled interior of the chloroplast is called the stroma, where the dark reactions of photosynthesis take place. Chloroplasts have several key features that enable their function in photosynthesis. Photosynthesis is the process by which chloroplasts convert light energy into chemical energy. This complex process begins when chloroplasts capture light energy from the sun. The chloroplast uses this light energy, along with water and carbon dioxide, to produce glucose and oxygen. The overall equation for photosynthesis shows that 6 molecules of carbon dioxide and 6 molecules of water, combined with light energy, produce 1 glucose molecule and 6 oxygen molecules. The organization of organelles within a cell reflects its complexity and capabilities. Prokaryotic cells, like bacteria, have a simple structure with no membrane-bound organelles. Their DNA floats freely in the cytoplasm. In contrast, eukaryotic cells have a complex organization with multiple membrane-bound compartments, each specialized for specific functions. This compartmentalization in eukaryotes allows for efficient division of labor, with each organelle specialized for specific cellular functions. The compartmentalization we see in eukaryotic cells brings significant advantages that enable complex life. This organization provides several key benefits that allow cells to function more efficiently. This compartmentalization enables cells to specialize for specific functions in multicellular organisms. For example, neurons specialize in transmitting electrical signals, using their compartmentalized structure to maintain ion gradients. Muscle cells are rich in mitochondria and specialized contractile proteins, allowing them to generate force. Plant cells contain chloroplasts that enable them to produce energy through photosynthesis. Complex multicellular organisms depend on this cellular specialization, which is made possible by eukaryotic compartmentalization.